I know even a smaller area of the DNA where it has to be in. If there was a recombination event and this person was not shaded, I would know for sure where's the disease gene. It'd be somewhere in these bottom three because this person is not affected. This person is, however, affected, and they still have the disease. They have the disease, and we know that they only have the beginning of this block. Therefore, whatever the disease gene is, it's located in that region in particular of that bigger block. For all of these, it's somewhere in here. That's where it actually is beneficial to us when we do see recombination there. Now keep in mind, if we had a bunch of recombination that would occur there, it wouldn't be haplotype block, because it would always get recombined, and we couldn't follow it through generation. So, once we've gone through the pedigree and we've determined, I know this is the haplotype block, we now want to then look at all the genes that are within that block. So what we do is we do an exon capture, which basically is we know what the genes are based on what? How do we know the sequences of all the genes? Everyone's favorite thing, Human Genome Browser. Right? There's a lot of browser stuff in this one and the next one. So we know what all those genes are, what they should be, and we can basically use exon capture where we can make things that are bind to that genetic sequence to help pull only those exons out. I can then only target those exons for those genes, and then I can sequence just those genes in particular, not all 20,000, but maybe there's only 5 genes in that block, I can sequence just those 5 genes. So I know what the overall sequence should be. Once I sequence it, I can then compare the sequence of this patient against the reference genome. And then wherever there is a difference, a mutation, hopefully if it's in one of those genes, then I can know, okay, this gene is mutated in this patient within this block. Therefore, we can assume that is the gene either fully responsible or partially responsible with this disease state. And you basically use you have things that bind to the actual exons, and they have a little magnetic bead on it, and then you can pull it out, and you take all the magnetic beads, remove them, then you just have your nice sort of sequences, then you can run it through the DNA sequencing, basically doing PCR with a whole bunch of it, and sequence it. Kind of like we talked about, right? Illumina. Keep in mind, what the Illumina machine with the massively parallel, you don't need to go back over that lecture entirely, but have a general, be familiar with what that machine is, what it's going to do in terms of allowing us to build up and then sequence that DNA. We can then look for, oh right here, look, there's a G there that based on the reference genome shouldn't be there. That may be the mutation that we're looking at in that gene, in that allele of that gene. So once again, that's for pedigree, and really that example was pretty simplistic. We're talking about dominant and recessive, where you have or you don't have it. A lot of diseases, though, aren't sort of dominant recessive. It's going to be basically a combinatorial effect, where we have a bunch of proteins, or a bunch of genes, that all play a role in a disease, and when enough of them are mutated, we then are going to have the disease state. What's the biggest example that Blake talked about for this whole lecture? Type 2 diabetes. Keep in mind for biochem, make sure you know type 1 versus type 2, because he expects you to know that on the exam, and if you don't, he's not going to tell you it. So type 1 occurs why?
You don't make it.
Autoimmune, which means do you have insulin or not? You don't have insulin. Don't have insulin. Good. Type 2, you have insulin, but it's insulin resistance. So you make insulin, but your cells the insulin can't really bind properly, or if it does, it doesn't bind at high enough affinity that we don't get the effects of insulin. Keep that in mind for biochem. What's up?
So are you saying you need to sequence someone's entire genome to find the SNPs and then do this exon capture?
So sort of. So basically, instead of sequencing all of the genes, that would take a very, very long time. It's much easier to work with VNTRs and SNPs. So before we look at the genes themselves, we want to look at and focus on what's traveling with the genes, which is the VNTRs and the SNPs. So once you do that, and I know just this region is where it is, I then go in and do exon capture of just that region.
How do you get the SNPs?
You have to sequence, you still have to sequence their DNA, but you're only looking at SNPs and VNTRs, because those are much easier to deal with than opposed to the actual genes themselves. If you're looking at the genes, there's 20,000 of them, and genes are really long and really complex. It's hard to get, you can do it, but it takes a really long time. But SNPs are either one base pair or two base pairs, or VNTRs are still just little regions. And they're much easier to do because what's the same next to every VNTR on either end? Between people, it's the same sequence, right? Once again, when we're talking about PCR with VNTRs, what are the primers attached to?
Conserve.
The conserved sequences directly next to the VNTR. You don't make a primer for the VNTR, that would defeat the purpose, because then you don't know how many times it's repeated. You're looking at what's right next to that VNTR and targeting that. So you're still doing a lot of DNA sequencing before, but focusing on the SNPs and the VNTRs. Once we have that, then we flip over and look at just the genes. Now we only have like 5, not 20 units. So yeah, good question. Once again, read the idea of recombination hotspots. That's where you're going to have a lot of recombination. The haplotype block is going to be where we're not going to have a very low chance of recombination occurring. But right here is a recombination hotspot, and right here is a recombination hotspot. Keep in mind, just because you have a recombination hotspot doesn't mean recombination is going to occur there. It just means there's a high chance it can occur there. So it's not saying it has to happen or not happen. In either case, it's not an all or nothing. It has a higher probability of happening if we are talking about a recombination hotspot, or if we're talking about a haplotype block, there's a low chance of recombination occurring right there. So here, once again, these are haplotype blocks. So this is one block. My first question is, does this add up to 100%? No. Why doesn't it? Are there only 2 blocks? Are there only 2 versions of a haplotype block in the entire population? No, there's way more than 2. And that's once again why I brought it up earlier. These don't have to add up to 100. If I wrote down every haplotype block at block 1, they would add up to 100. But blank only gave us 2. So it's not saying these are the only 2. Let's say you have the orange block, the green block, you may have a red block, a blue block. Once again, within each of those blocks, everything is the same. However, what we're looking at here, we're just focusing on 2 for block 1. What we see is that in the population in general, 76% of us in this room have this block right here. Another 18% have this block down here. Once again, it travels together. So in the population at large, 76% have orange. The other, whatever it is, 18 have green. So that means that if I have a G, assuming no recombination within that block, if I sequence these blocks in all of you, and let's say I'm looking at the people with the orange one, if I found a G right there, usually we don't want to just use one, but let's say we did 2. If I found the G and the C, I can say pretty certainly what all these other SNPs are going to be. Because I know what one of them is in the block, and everything in the block travels together. What's the whole point of this again? Is I want to look at this, but also look at, let's say we have the D gene in there again. That D gene also travels in here, and whatever allele is there will always travel with it. So let's say in the big orange, it's a big D. And let's say in the green too, it can both be big D. But then in another one, the red one, it's little d. All right? So that's the general idea there. And what's occurring in between here with these lines, this is the recombination hotspot. So that is where there's a high chance of recombination occurring. So if I tell you, you have a patient, and in this block 1 they have this haplotype block, what does that tell me about this block right here in that patient? Can I tell you anything of certainty? No. I have no idea what block is in that. I have no idea which haplotype block they have at block 2 position. I have no idea. Because recombination could occur there, or maybe it didn't occur there. We don't know. So whatever we know about, any information about something within the haplotype block only pertains to that block in particular. Everything in here moves together, everything in here moves together. But these 2, I have no idea if they're moving together or not, because recombination hotspot is right in between there. What's the term here? There it is, the linkage disequilibrium. Basically, that just means that it travels together. All right, that's the sort of how I think about it. That term is just kind of confusing for me. Linkage disequilibrium means that they are linked together. Like I think the actual definition, I'll write it down too. Non-random association of alleles at different loci in a gene in a given population. Like I said, they travel together. All right. Linkage disequilibrium means that if something is within a block, one block itself, it is basically in linkage disequilibrium. So they're linked together. They always travel together. However, these two, this A right here and this C right here, are not in linkage disequilibrium. Why not? Because they're in different blocks. And there's a recombination that possibly could be occurring right here. And in some people, that recombination will occur when you make your gametes. Other people, it won't. Because it's basically doesn't mean you have to have the recombination there. It's just a high probability that it will occur. And once again, so what we end up doing, we're basically looking at just this block, and now we're zooming in here. Keep in mind, these blocks are fairly large. They're talking about sometimes they are kilobases big. They're very large amounts of DNA. The majority of that DNA is going to be the same. It's going to be not changes in SNPs and alleles and VNTRs. However, there are going to be unique places where there will be differences, and that's what we highlight here are just these individual SNPs. Once again, the whole point of doing these GWAS studies is to, because it's easier to work with VNTRs and SNPs, identify those first within a block. Once we do that, then look at genes within that block, and then look at differences in those genes. Then we sequence test those genes on their own, and then we can get a good idea of what the issue is going to be. So once again, here, just understand the actual chart. Obviously, this is the amount of recombination activity. So here's obviously a hot spot. It's where there's a high amount of recombination, whereas things in here, very low recombination. It's never actually zero, it's pretty close to zero. That would be a haplotype block, where there is overall a very low chance of recombination occurring. This is what we've spent the whole time talking about. I can sort of talk about it out loud. The idea in the beginning, how do we first see, oh, people with Huntington's disease have it's the Huntington gene that's what's screwed up. What they did is you identify the candidate region. How do we do that? Look at the SNPs, look at the VNTRs, at all those haplotypes, in particular within the haplotype that is common to all the diseased patients. So whenever we're doing these things, we can't just use the diseased population. I have to compare my diseased population to my normal population. So once again, why we can't start at the genes at the beginning. We have to do a bunch of sequencing of genes in the diseased population and the normal population. Keep in mind as well, we're talking about things that are non-Mendelian, so these qualitative traits or qualitative diseases like diabetes. It's not just one gene that could be. Maybe in this family, it's this gene that's screwed up that leads to diabetes. But in a different family or in a different population entirely, it's a different gene that's screwed up. And usually it's not just one gene, it's a combination, it's a combinatorial effect. We sort of have to keep that in mind as we move forward. When we do that, do clones of the DNA, identify just those genes there, exon capture, then look at that, sequence those genes, look for the mutations, compare it to the reference genome, and then we can say, all right, that gene is probably the one that's screwed up, we can assume. So now that I've identified that gene, I can then take this same thing and now apply it to larger populations. Instead of dealing with a family, I can deal with like an entire population of people. Why? Because now I've narrowed down what gene I need to capture and sequence. In the beginning, I didn't have that. That's the whole idea of doing it. Keep in mind, too, knockout studies are going to be important for this as well. Where would we probably add a knockout study? Like, after what step do you think you would do knockout studies? If you're doing a knockout study, do you know what the gene is? Do we need to know what the gene is to knock out, to do a knockout study? Okay, yeah. So we can't do anything, basically, until we've identified what the actual gene is. So whatever the unique genes that screwed up, from there, before I then say, okay, let's look at the whole populations to find it, double check myself, or to understand more about it, I can then, let's take this gene in mice and screw it up and see what the effects are. If it's similar to the actual disease state, That's another way of saying, okay, good, we know what it is. In science, you don't want to say, I think it's this, we'll just spend all this money on it from here on out. You want to always make sure, without a doubt, what you're focusing on is what actually is occurring. So once again, you've got the quantitative traits versus dichotomous traits. Dichotomous traits are basically you have or you don't, like big A, little a. For these quantitative traits, it's basically due to a combination of different genes. And maybe in some patients, all their genes are screwed up, they definitely have the disease. Or maybe in other cases, only 2 out of the 7 genes are screwed up, but they're still, just those 2 of them being mutated will still cause the disease to have you, makes you have the disease. That basically is represented here is just the threshold. So for diabetes, what is the variable we're looking at between disease and non-disease state? Glucose levels in the blood, right? So if you have 2 high glucose levels in the blood, that basically is what's going to be down here, is your glucose levels. And in terms of the higher you have of that, if you're over, I don't remember what the actual value is, but if you're over whatever that value is at resting blood glucose levels, then you have type 2 diabetes, or even just diabetes in general, but in this case we're talking about type 2 diabetes. And that would be the group that's within this range up here. For diabetes, it's much more this way than that way, but it depends on the scale. But yeah, so keep in mind, for those quantitative traits, it basically just means there's a large quantity of genes. That's the whole idea. Large quantity of genes that are going to be important for determining whether you have the normal or the disease state. That's the example here, where it's not just having, this is just a dichotomous trait, where you have one loci. One loci means like one location of the chromosome for one gene. You either have little a, big A, or combination of the 2. And that's your frequency. However, as we begin to add more and more genes for a quantitative trait, we approach more of this bell-shaped curve, because now we have so many more possibilities that we can have going forward, right? Because now we have more genes. In this case, we've got the A gene, the B gene, and the C gene. So the more genes we add that are affecting this trait, the more we're going to approach this bell-shaped curve. And then once again, depending on whatever the values are going to be, we can say that if you're above 110, whatever this is the disease state. Whereas everyone down here is normal, not affected. So you can still have, in some cases, you can have one mutated or one basically bad allele for the gene. But if you're talking about a trait that has like 15 genes that are at play, one of them being the non-wild type or being the disease state may not put you over the edge for whatever it's going to be. It's usually a combination of more than one that has to be occurring. It just depends on the different genes that we're talking about. So one way to tell, I think this is when he goes into, yeah, cool. So looking at diseases a little more, we often want to see, is there a genetic component to the disease? So does it run in families? Does breast cancer run in families? Does diabetes run in families? That sort of thing. And the easiest way to do that is just look at this, pretty simple. Look at the family and compare that frequency to the general population. So if in the general population, 2% of people have this disease, all right, that's great. However, within just one family, if it's also 2%, is there a heavy genetic component to it? No, because it's the same as the general population. However, what happens if we're looking at a family and we see this, where almost, I think about half or so, have the disease. However, in the general population, we're at 2%, then yeah, there's probably there's a genetic component associated with the disease. Because the frequency in that family, that's what all this photo is showing us too. In this family, there's a much higher amount more number of people in that family have the disease compared to the general population. Now, what could be another reasoning for this? Does it have to be genetic? Do I know for 100% it's genetic? What else could it be? What happens normally with families? You live together, right? So it could be environmental if they're experiencing or living in the same environment. However, here, this is like first-degree relative, so this is usually, you don't just use it for one family, but usually the family and talking about maybe cousins, nephews, or people that are still within that overall genetic sort of family, but even if they don't live with each other, that's another way for us to make sure it's more of a genetic versus an environmental. But keep in mind, it's like that nature versus nurture thing, so just don't always say it's always this, never that. And we look at here for some diseases, we look at the fact that we have increased risks for the sibling having it.